I'm going to jump right in and I'm going to give you essentially a historical perspective of my role in this field over the last 25 years. I'll just run the whole dynamic here. So really what I'm showing you here is the timeline of BRCA1 and BRCA2 research. And way back in 1990, 1992, when Mary Claire King discovered that there was an actual locus for breast cancer, so a locus being a region where there was a gene located, but the gene was unknown at the time. And then we embarked upon, a 4-year search for that gene. And that was successfully identified in 1994. And then very quickly, we had learned so much from that 4-year odyssey that we had BRCA2 within 1 year after that. And so it all moved very quickly at that point. And then the next few years were spent trying to understand the biology of the proteins, these new huge proteins that had just been discovered that we didn't know anything about. A lot of people trying to do experiments in that area. And there were some key studies that came up here, such as the value of prophylactic mastectomies, the value of prophylactic oophorectomies, and Susan actually led a number of those studies back then. And then we had the advent of understanding the DNA repair activity of these proteins, and we had PARP inhibitors identified for the first time. And of course, we've worked for years now, many clinical trials on PARP inhibitors trying to show their effectiveness for breast, ovarian, and now prostate and pancreas as well. And then we kept going and we moved into genome-wide association studies, trying to identify these common genetic elements that combine together in what's called a polygenic risk score, could be valuable as a risk factor for predicting who's at elevated risk of cancer. And so I was involved in a number of those studies. And then we had to turn over the patent, I think about 2015, if I have the right year. 13, she has it memorized. So there was a turnover of the patent, which then opened up the testing to many different companies to come in and more global exposure to testing. I think that was the advent of all that. And then we've continued with the PARP inhibitor studies here. And as we move into over the last few years now, just more recently, it's been trying to understand the additional breast cancer genes beyond BRCA1 and BRCA2. What other genes are involved? What's their level of risk? How can they be treated? There's a lot of treatment work going on as well. So that's sort of the timeline of the field in a snapshot. What was my role? Well, I started as a postdoc with a person called Barbara Weber, who was actually here at the University of Pennsylvania. So I was a postdoc here for 4 years. And I worked with Kate Nathanson. We were fellows in the same lab. And then I left shortly before Susan joined. But Susan also joined and worked with Barbara before she left for industry. And this guy in the middle here is Francis Collins. A number of you will know that name. Ex-director of the NIH, I guess, who had a major role in the COVID response, et cetera. So he's done tremendous work over the years. So I've been in this from the very, very, very beginning, the search for the BRCA1 gene, all the way through to the current day. So here's an updated slide that was provided to me courtesy of Claire Turnbull, who's from the UK. She works in the Wellcome and Genome UK, where she showed the various breast cancer genes. So what you're looking at here is how common are the mutations in the general population, and then vertically, how much risk. So we think of BRCA1 and BRCA2 as very high-risk genes, way up here, relatively common in the population. So about 1 in 300 people have a mutation in one of these genes. But then there's all these other genes that we like to talk about, and we have PALB2 now, we think, is another high-risk gene. CHEK2 and ATM are often talked about, and they were talked about a lot this morning. These are sort of moderate-risk genes, more moderate-risk genes that used to be thought as just ovarian cancer genes. Now they're breast cancer genes too. And a few other syndromic type genes that are incredibly rare, but nonetheless do predispose to breast cancer. And then over here, we have all these risk SNPs, these genome-wide association study products that lead to these polygenic risk scores that I was talking about. So this other risk factor. You can see that their level of risk is incredibly low. It's really very low for the individual SNP. But when you pool together 313 of them, which is what we currently do, it really has a major impact on risk, and it's very useful. So how did we really get this whole field moving to understand BRCA1 and BRCA2, to understand genome-wide association studies and these common polymorphisms, to understand the new genes that I've just talked about? Well, we formed consortia. So it's all about team science. These are people from all over the world who joined into consortia, and these are called the Breast Cancer Association Consortium up here. And then SIMBA, this is a snapshot. This is what we like to do. This was during COVID, so it was all virtual. So we took a snapshot of all the people on the virtual call just to show all the people who were there. And I think there's something like 50 countries involved, 150 people attending. These are really wonderful consortia where we join all our patient data together to get massive numbers, because this is really a statistical game. You just need massive numbers to do some of these types of studies. Okay. So I'll start with the genome-wide association study. We've heard some of that already today, but I just want to touch on that because I was heavily involved in some of these original studies. Where are we today? Well, we have 313 individual SNPs, or single nucleotide polymorphisms, that contribute to risk. But as I said, each one has a very minor effect on risk. But we can combine them together in what's called a polygenic risk score that actually now is a very strong risk factor for breast cancer. And so I was involved in this right from the very beginning. This was formed in about 2012, this consortium. And we've gone through multiple phases of GWAS, and I was lucky enough to actually lead the original studies on ER negative, so estrogen receptor negative breast cancer and triple negative breast cancer in the setting of genome-wide association studies. But again, it's a team environment, and so a lot of these types of studies are shared out amongst the various investigators. And then on the bottom we have the SIMBA group, which is the BRCA1 and 2 only. And you can see the massive number of BRCA1 and 2 mutation carriers that have been collected through that consortium. Huge. And again, it's worldwide, everybody pooling in together. And again, we have a polygenic risk score for that. And I was lucky enough to lead the studies on BRCA1, the original GWAS studies on BRCA1. And then Ken Offit actually led, from Memorial Sloan Kettering, led the studies on BRCA2. And we worked very closely together to do a lot of that work. So I like to feel like I was the initiating force behind some of these things. Other people have taken it on and have led the effort since then. But then we have a new study called Confluence that's led by the National Cancer Institute, which again has pulled all these people back together again and is doing another massive genome-wide association study to further extend our knowledge of these risk factors. And so this is just to say, how do you use a polygenic risk score? So what are you looking at here? Well, you're looking at, if you think of the polygenic risk score as 1 to 100%, it's how much of the polygenic risk score you have along this line. And then it's how much risk of breast cancer you have on this. So 1 would just be normal everyday people. So if you're above 1, you're at elevated risk. If you're below 1, you're at lower risk. And you can see over on the tail here, the people with the top 10% of the PRS have a substantially elevated risk of breast cancer. But there are also people who have a substantially reduced risk of breast cancer. And this is potentially the utility of this model, right? You can find people who go up in risk and people who go down in risk relative to the normal people. So this is really quite useful, and we talked about this the last couple of days, we're still doing studies trying to figure out how to implement this in the clinic, but it's coming, and it's coming fast. Okay. And then this is to show that we can also extend this to the mutation carriers. So the previous study was just all breast cancers. Now it's the mutation carriers, and you can see what we're doing here with the peak. If we take the PALB2 peak, we're saying, well, the average PALB2 would be a line right down the middle here. But in fact, with this distribution across, you can see some PALB2s that have low risk way down here, some PALB2s that have very high risk way up here. So there's this distribution, this separation across the mutation carriers that again will be very useful for risk prediction and risk assessment. We've now talked about these guys, so now we're going to move over to these genes. So when we think about breast cancer, many times we talk about high-risk patients. And that's generally people who are diagnosed at a very young age, people who have a family history, a number of other measures that we use, multiple cancers, so not just breast, but breast plus a few other things. So we tend to use this as a flag to say you should genetically test this individual. But then there's the general population. There's all the other people who got breast cancer that don't have those characteristics. They have no family history. What are we going to do with these people? So the way we've approached the genetics of breast cancer is to try and understand both sets independently. We really see them as two different groups of people. So I hope you like tables and numbers. We're going to be looking at a few. Trying not to bore you too much, but these are the genes that we like to test for. These are the ones that we know are driving breast cancer, and you have your BRCA1 and 2. And the number here means the fold increased risk over the general population. So you're 5-fold increased risk if you have a BRCA1 mutation. You're about the same fold increased risk if you're a BRCA2 mutation carrier. So you get the idea. Population risk is 12% of all women will get breast cancer. You're 4 or 5-fold above that, right, if you have a BRCA1 and 2. But you can see some of the other genes are a lot lower. They're only maybe 25% lifetime risk instead of a 60 or 70% lifetime risk. So we spend a lot of time doing studies, and way back in 2017, as you'll note on the bottom here, there really wasn't any information. There were many, many, many small studies that had been done, but we had no stable estimates of the risks for any of these genes. Even for BRCA1, it was a little bit messy. So this is a study from Ambry Genetics, which is one of the genetic testing companies that we've collaborated with extensively over the years. And they were nice enough to just give us all their data and let us do all our genetic analyses with that. And what we came up with using 50,000 breast cancer patients and 50,000 controls, I mean, nobody had those numbers previous, so this was really a windfall at the time, and we were able to come up with these stable estimates. So that was a fairly significant paper in 2017 where we finally gave people tight estimates for most of these genes that they could work on in the clinic. But we also found at the same time that some of the genes that had been thought of as breast cancer genes turned out not to be breast cancer genes. We had seen a few families with these mutations, so we made the assumption that they were breast cancer drivers. Turned out they weren't. And the data is actually right here in particular. So for instance, the NBN gene, which was a very well-known breast cancer gene for a decade or so, we did a study and we found that it was really low risk, barely above just regular non-risk, right? So it just pushed it up a tiny, tiny bit. And other studies have validated that. So in fact, a number of these genes ended up being considered as not breast cancer genes, but interestingly, they're still on the testing panels. And that's just for convenience for the companies. They have their technology set up. They don't want to start pulling things off. But it does complicate matters for the genetic counselors and physicians who have to deal with the data. Okay. And then we went through a whole series of studies. So we had risk for high-risk breast cancer patients. We were able to give them numbers. What else could we do with that? Well, we looked at subtypes. So subtypes are breast cancer can really be broken down into a few different subtypes of the disease. We always talk about breast cancer as 200 different diseases, but we can break it down by the estrogen receptor, progesterone receptor to some extent. And we come up with these 4 groups, and then we looked at each of the genes by each of the 4 groups. And you can see that sometimes there are tremendous differences. So if we look at BRCA1 as an example, we always think of BRCA1 as an ER-negative breast cancer or triple-negative breast cancer driver gene, right? And that's that purple one way out here, really high risk. But interestingly, it does cause the other 3 subtypes of breast cancer a little bit as well. So there is some risk for the other ones, but it's clearly different between the two groups here, or the 4 groups. And you can see that there's other genes that are like that too. CHEK2 doesn't cause any triple-negative breast cancer, but it does cause estrogen receptor-positive breast cancer. And there are lots of stories like this on this chart. I'm not going to go through all of them, but you get the idea that the subtype of breast cancer actually became quite important. And then we were able to move ahead and look at race and ethnicity, and that was mentioned earlier. And the BASR is putting a lot of effort into this kind of work as well. In this case, you can see some of the numbers there. We had 57,000, so 77,000, 78,000 individuals with breast cancer. So again, huge data sets available from these collaborating companies that we were able to break down some of the subpopulations here. And again, show that there were some differences in some of the populations. African Americans tending to have more triple negative or ER negative breast cancer, so their risk appears higher in this setting. But then we thought, okay, well, these are all the high-risk patients. But a question was then posed by this paper here, the Beich paper in 2018, which actually said that when we test all breast cancers, we actually find mutations, 50% of the mutations that we find in BRCA1 and 2 and some of these other genes are in people that don't actually have a family history. So that's what I was talking about earlier about a general population. So 50% did not have a family history in any way, so we would have walked right over the top and never tested them, okay? So we did a study using that same population that I was talking about earlier, and we cleaned it up a little bit. If you notice up on top, they had many different genes here. Some of these are not involved in breast cancer in any way, so it was kind of a dirty study to some extent. So we cleaned it up and focusing on the key genes. And it ended up that we were actually missing 35%, still a lot. But in fact, we were finding nearly all the BRCA1s and 2s. The ones that we were missing were these other sort of moderate risk genes, but still important for the patients. If you happen to have one of those mutations, it's really quite important. So I think this was a bit of a game changer paper as well, in that it really showed to everybody that we're missing a big chunk of these people, and we need to be thinking about people in the general population with breast cancer. So that led us into population-based studies. So this is the general population of breast cancer, not selected in any way for early onset or family history, etc. And we developed what's called the carrier study. And this is where we reached out around the United States and we found some of the large cohort studies. So the American Cancer Society had a couple of big studies that they were able to provide a lot of DNA samples to us. The multi-ethnic cohort, the California teacher study, a number of these really well developed and really well understood cohorts that had been in existence for 30 years. So they would enroll 100,000 people at the very beginning and follow them over time to see who gets breast cancer. And that's a really elegant epidemiological design if you can follow people through time and see who gets the disease over time. So we were able to get samples from all these people and mutation test them in our lab ourselves. 90,000 people it ended up being, which is a huge task. It took us 4 years to do that work. And here are the results. So what are they showing you? Well, if we can just focus on the ones with the tall blue peaks, it's those same 5 genes again. It's BRCA1 and 2, it's ATM and CHEK2, and it's PALB2. So we think of BRCA1 and 2 and PALB2 as the high-risk genes. ATM and CHEK2 is the moderate risk genes. And these are the 5 that most people focus on. And again, if you just look at the frequencies, these 4 are about 1% each, and this one is a little bit lower than that, maybe about 0.5%. So between the 4 of them, you end up with about 5% of random everyday breast cancer patients who have mutations in one of these 5 genes. To most people, that was a surprise. That seemed like a lot, that 5% would be walking around with mutations without any restriction on age of onset or family history or anything like that. And then at the same time, if you look at this sort of orange bars, these are what we call controls. So unaffected people that we kind of match up with the affected people just to see if there's differences. And sure enough, there were a lot of mutations in that group. And it turns out that 2%, almost 2% of women, no breast cancer, walking around on the street, 2% have mutations in these genes, which I think is a very high number. But some people would say, well, that's a very low number. So it comes down to opinion. But it still seems a lot to me for people who are unaffected at, let's say, an average of 50 years of age. Okay? We did then realize that, you can calculate all these statistical numbers. We can talk about odds ratios and relative risks and hazard ratios and all these things. But what does it actually mean in the clinic? It's not that useful. But what does matter in the clinic is the cumulative risk. So what that means is your risk by age. How much risk of breast cancer do you have as you go across your age spectrum all the way out to age 80? And you can see the BRCA1s and 2, sorry. You can see the BRCA1s and 2s that run up to around 60%, which is in keeping with other studies. It's a little bit lower here because, again, we're looking at the general population, not high-risk patients. And then PALB2 was quite high, coming in around 35%, which matched some other studies that had been published. And then ATM and CHEK2 on a different scale here, coming in around 20%. So our numbers were actually consistent with what had been seen in high-risk patients, but a little bit lower because it's the general population. There was no family history here. So that made a lot of sense. But it's useful in the clinic because now people can look at, well, what happens at age 40, what happens at age 50, what happens at age 60, and you have numbers as a result of that. And then we were able to take that carrier study and put it together with a study called Bridges from the UK. So Carriers was a US-based study, this was a European-based study. And we merged the two together to look at the big numbers. And this is sort of a little bit too complicated, I think, but basically those are their numbers above and our numbers below. And what you can see here is that the risks are similar, right? These main genes are all elevated in both studies, which is nice to see some validation there between 2 studies. Some animation. And then here's the 2 thrown together. So there's an estimate of what the risks are. And so what you would say is these numbers here are sort of the final take-home message from these 2 huge studies that are now combined together. And they're very solidly putting these 3 genes into the high-risk breast cancer category. And so the same is true of these intermediate genes that I've been talking about, the ATM and PALB2. Again, we get very similar numbers. I'll just keep going. So again, similar numbers again. I'm just going to keep pushing through this if you don't mind. The point there was that we were both finding approximately 2% of those unaffected people were having mutations. So it wasn't just something funny in our carrier study. It was seen in the Bridges study as well. So everybody's seeing 2% of unaffected women with mutations. And then we were able to start reaching out into other populations. So this is where we started with an African American study. And we were lucky enough to have a large collection of about 4,000 breast cancer patients and about 5,000 unaffected African American women that we could match up and do the studies. And what we actually found, we won't look through the numbers, but what we actually found is that the same genes are driving breast cancer in African Americans as in whites. And the amount of risk is about the same. So this was really kind of gratifying for the community because it means that we can use the same testing panels for all the different populations because it's the same genes and it's the same risks, right? So that was important to see. And then we were able to put together these lifetime curves that I talked about earlier, and this is for African American women, lifetime curves. The risks are a little bit higher because they have more triple negative breast cancer. That's just a bit of a statistical artifact, but it's very much in keeping with what we saw in the white group. And then we decided to look at older women. And Susan played a major role in this study. The question was, we tend to test younger women, test women up to the age of 60 or 65. Sometimes actually the NCCN cutoffs are right now are 50 or triple negative breast cancer. So a lot of women over the age of 65 with breast cancer are just ignored. They're not tested. And we said, well, why not? You know, women are living to 82 years of age right now. That's the average survival. So why not look at these older people? So we did. And we find that there's actually a lot of residual risk still there. There's a lot of people with mutations in that older group that could actually benefit from some sort of intervention. I will admit, though, in this study, we did find that the effectiveness of this study was really in the estrogen receptor negative, triple negative group, and less so in the estrogen receptor positive. So if you're an older woman developing estrogen receptor breast cancer over the age of 65, probably only 1% will actually have mutations. It's not really that effective to mutation test that subgroup. But if it's triple negative or ER negative breast cancer, you should probably test them because it's much higher, and then they have the benefit of PARP inhibitor treatment. Okay? And then we went back over to subtypes. So now we're talking about the general population. I talked about subtypes for high risk. Now I'm talking about subtypes for the general population. Again, very similar numbers, again, showing these differences across the different subtypes. So just very consistent data. Here's a graphical view of that. So it's really nice to see that the general population and the high-risk patients really are showing the same genes and similar risks, but the general population is always attenuated or a little bit lower, which makes a lot of sense. That family history effect has gone away and it's sunk down a little bit, but still very clinically important. And then more recently, we've published a study on contralateral breast cancer. So this is the idea of that if you have a breast cancer and you have a mutation in one of these genes, what's your chances or your risks of having a second breast cancer on the other side, on the other breast? This is really important. These questions come up all the time in clinic. And actually, there were no real solid numbers for mutation carriers for this kind of information, even to last year. There wasn't a lot of information. So we did a big study in our carriers group. Again, Susan was very involved in that. And we were able to come up with estimates showing that several of the genes are really major drivers. BRCA1 and 2 do have a high risk of a contralateral disease. And it looks like some of the others don't either. But if you go to the different types of breast cancer, so in particular PALB2, which we think of as a high-risk gene, it only has an elevated risk of contralateral breast cancer if your original breast cancer was ER negative or triple negative. But it doesn't seem to be the case when your original cancer was ER positive. And we haven't quite figured this out at all. It could be that prevention treatment with tamoxifen or aromatase inhibitors, or even treatment of the original tumors with aromatase inhibitors may have prevented the onset of these second cancers, and that's why the risk is lower. So we're doing some studies to try and figure that out. It's really quite provocative information. And so this is what some of the graphs look like. What you're looking at there is This is the general population contralateral breast cancer risk, and then this is what happens when you have a BRCA1. It's much elevated. Same for BRCA2, and I didn't show the other genes. So now I want to move from loss of function mutations, which is what I've been talking about the last few minutes. So these are knockout type mutations in all these different genes. Now I'm going to start talking about what are called variants of uncertain significance. And these are really well known to people in the business. What happens is that when you undergo a genetic test of your DNA, sometimes they find these events, these changes in some of these genes, and nobody knows what they mean. The testing lab doesn't know what they mean, the molecular biologists don't know what they mean, the patient surely doesn't know what it means. And so there's just confusion all around. And unfortunately, as a result of it, so you have a genetic diagnosis, which is, hey, I have a variant, but I don't know what it means. And so you can't really benefit from all the screening, prophylactic surgeries, and treatment options that someone with a real BRCA1 or 2 mutation has. You've got this unknown thing that you just don't know what it means. And so my lab has really focused over the last 5 to 10 years in really trying to figure out what all these things are and which ones are important, which ones are actually driving your breast cancer, and which ones are just benign, random genetic variants in the background. So in order to do that, we formed what's called the Enigma Consortium, yet another international consortium. And again, this is worldwide. You can see all the flags and where they're located. And again, it's about, I think it's about 100 groups now that combine and come together every year and share data, share information, and we do routine conference calls all the time to share our information and try to advance this field. So I'm going to talk about BRCA2 specifically in this context, in the context of variants of uncertain significance. And we focus in on this area here. This area turns out to be the only region of the entire gene that has what we call pathogenic or disease-driving missense mutations. Missense are single little amino acid changes. So they're only found in that region. They're not found anywhere else across the gene. There are many changes, there are many missense variants of changes found all the way across the gene, but none of them have ever been shown to actually be risk drivers. It's only the guys down here. So we embarked upon this. We actually do functional studies in the lab. And these are done in cells. And we use a readout here. And what happens is that when the cell undergoes DNA repair, the cell actually turns green. And you can see it under a microscope, and you can actually count the number of green cells. That's how it works. And so on this graph, I'm showing you 450 different variants. The amount of DNA repair is here, from none all the way up to a lot. Okay? And you can see that there's many, 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 many variants in this DNA binding domain that seem to have quite a lot of DNA repair activity. So they're actually normal, right? We use the term benign, neutral, normal. We throw these terms around all the time. But all these guys look normal. But if you look here, there's a whole bunch of them that are much lower. They're way down low. They've lost a lot of the BRCA2 activity because of this tiny little change. What we found with this functional assay is that it actually has 100% sensitivity and 100% specificity for pathogenic missense mutations in this region of BRCA2. So when you see a result like that along the bottom, you're fairly certain that this is a knockout mutation. It's really destroying the function of the protein. And interestingly, there's 137 different variants here. So if you go into ClinVar or ACMG or any of these sort of public databases and you look it up and you say how many missenses in BRCA2 are actually confirmed to be pathogenic variants, there's only 22. And so with our functional assays, we have 137 that we know knockout activity. Now the question is, can we take that activity and combine it with other information to actually formally clinically say that it's a pathogenic variant, right? But you get the point here. The functional assay can potentially massively increase the information content on all these variants. So we did that. We actually teamed up again with Ambry Genetics, our friends at Ambry Genetics, and we did a study where they gave us 154 missense variants that they called VUS, variants of uncertain significance. Didn't know anything about them. We ran our functional assay, combined it into what's called their ACMG, or American College of Medical Genetics mechanism, series of rules that you can combine all these data sets together. And we ended up classifying the vast majority. Only 22 of the 154 remained unclassified. So we classified 86% of the missense variants in this study by just doing this functional assay and combining it with other data. So that's powerful. That says to you that good functional assays, well-calibrated, well-validated assays, can really shift the goalposts in this type of work, right? And start to classify lots of missense variants. And we actually did another study with another company called GeneDx. And in that one, we took a whole different set of variants in BRCA2, and we got 92% ended up being classified. So between the two, when you average them all together, there's a very large number of newly classified variants. Over 90% of the missense is taken care of in that setting. Still a few remaining, but the vast majority taken care of. So as a result of that, we formed what's called the Variant Curation Expert Panel. This is a mouthful. The Hereditary Breast and Ovarian and Pancreatic Cancer Variant Curation Expert Panel, HBOP-VSEP, okay? And this is run by the NIH. They have a body called ClinGen, and their whole goal is to find methods to clinically classify the importance of variants in many different disease genes for many different disease states. It's not just breast cancer, okay? And then they try to come up with these rules that can be applied to all these different genes. We formed this committee to make rules specifically for the breast cancer genes and the ovarian cancer genes and the pancreatic cancer genes. And we didn't do prostate because we didn't know so much about prostate when we first formed this, but we may co-opt those guys at some point. But you can see it's from all over the world, and we had clinicians, we had epidemiologists, lab-based researchers, genetic counselors, everybody you can think of is on this committee. Some of the people in this room are on this committee. And so it's been very productive. We've now gone ahead and made rules for ATM, PALB2, BRCA1, BRCA2, and we're working on another gene called RAD51C. So all these rules are about to come out in the publication literature, and they will allow other people to try and classify all these variants with very strict methodologies on how to go about this. So this is really, this is all voluntary, by the way. None of these people get paid to do this. They're just doing it because they think it's important. So all that work is great, but we spent 10 years doing 450 variants. That's not fast enough. There's 4,000 variants in BRCA2. We only did 450. So we'll all be retired by the time we get the rest of them done using this methodology. So we decided to move into newer technology, and this is CRISPR-Cas9 based. So the idea here is that you insert the mutation into the genome of a cell and then see how the cell behaves as a result of that. But you can do this in massive parallel ways. In other words, you can do this for all the mutations in a particular part of the gene all at the same time. So you can take 1,000 mutations and stuff them all into the cells all at the same time and see how they read out. And we use high throughput next-gen sequencing to actually get the results. The idea being that the nasty mutations are killing the cells. The okay mutations, the neutral ones, the cells are surviving. And so you get more DNA from the surviving cells and less DNA from the dying cells, right? And you can just kind of make ratios. And this is unpublished data now from the work that we've been doing on BRCA2. And what we've done here is taken part of that DNA binding domain that I talked about, the first half of it actually, and this is the data from the whole thing, from that part. And the red bits here are the known pathogenic, the known deleterious pathogenic variants. The sort of light blue color, gray color here are the known neutral variants. And you can see that there's pretty good separation of these guys. They overlap a tiny bit in the middle, but for the most part they separate. And then the blue ones are all the missense VUS that I'm talking about. And I think you can see that there's a suggestion here that you could actually very clearly identify all these missenses over here as being the same as the deleterious ones, and then potentially classify them. And again, you've done thousands of them all at the same time. So there's actually, I think, 3,000 variants on this screen alone that you're looking at. And so here's just exon by exon some of the results that we have. You can see how well it's worked. So really high scores in terms of sensitivity and specificity for nasty ones versus neutral ones, suggesting that this is really going to work well. And you can see that 2,592 variants that we've evaluated in this way. And so from those 2,592 SNPs, we end up with 435 missense variants, so VUS, that we now say are deleterious based on this experiment. So before I took 10 years to do 450 and it came up with 137. Now in 6 months or a year, I've done all these guys and come up with 435. Again, the goalposts are shifting. So this work is ongoing. This is where we compared it with some other DNA repair activity that we had, and there's a really nice correlation. So everything seems to be working properly. And now, instead of just a single cut point, what we're trying to do is actually get more refined. We want to be 99%, not 94 and 95, right? We want to be really certain of our results. So what we're actually going to do is set thresholds here so that we have an uncertain zone in the middle, and that everything to the left, we're really confident it's bad, it's deleterious, and it's pathogenic. And these guys to the right, we're really confident that they're neutral. We don't want to make mistakes and give wrong results to patients, right? So we will live with the fact that we'll have an uncertain zone in the middle in order to be sure of the other ones. That's really important. So that's what we're working on right now. And then as part of that, we found a series of BRCA2 variants that we call hypomorphs. So this is this uncertain zone that I was talking about. It turns out from all our studies that we've identified 70 variants in BRCA2 that have partial activity. They're not knocked out, they're not normal, they're partial, they're in between somewhere. So why is that important? Well, because if somebody shows up with a BRCA2 mutation, you look at them and you say, you've got 70% lifetime risk. There's suggestions of prophylactic mastectomies, et cetera, right? But here's a group of people who have lower risk. So they've got lower activity, intermediate risk. They actually behave more like the moderate risk genes like CHEK2s and ATMs. And we generally don't counsel prophylactic mastectomies for those patients. It's more of a screening methodologies with MRIs and mammograms, okay? So the concern here is that these patients, once these variants are identified, the physician might be inclined to say prophylactic mastectomy might be a good idea here, but in fact we know they're only at 25% lifetime risk. So you have to dial back. And so what's going to happen in the field is that we're going to have to come up with whole new guidelines for this set of patients. They are BRCA2 mutation carriers, but they're different BRCA2 mutation carriers. That's the point. And this is a really important observation. There's only one BRCA2 variant in the literature known to be a hypomorph. We've got 70 in this study here, and this paper's under review. So it's a bit of a game changer. We also validated that with CRISPR as well, because we wanted to show with two methods that it would be the same. And sure enough, here's this group of hypomorphs sitting right in the middle, as they should be. And then we treat them with drug, with PARP inhibitor, and they shift over to the left. They start, when you hit them with PARP inhibitor, they look actually more like the deleterious pathogenic variants. That's really interesting because what it says is that you may have a hypomorphic variant and you may be at lower risk, but you'll still respond to a PARP inhibitor if you get a tumor. And that's really quite useful information. And this is where we just proved, we lumped them all together, we looked at them in cases and controls, and what we actually found was that the hypomorphs are in between. So these are nasty known pathogenic mutations, 8s and 9s. These are neutrals, 1, no change in risk. And then our hypomorphs are sitting right in the middle at about 3. So that's for breast, and then we do the same for ovarian. So we've actually shown statistically that this group of hypomorphs are in between, and they're very different to your average everyday BRCA2 truncating mutation. And so we've also modeled out the lifetime risks, so that will be quite useful to the physicians and patients and counselors when this all comes out. So we have hypomorphs, they do exist. It's another category of BRCA2 mutations that we're going to have to deal with and come up with new guidelines for how to do risk assessment and risk management for these patients. And so let me summarize then everything that I've talked about. There's a lot of testing going on, both the high-risk genes and the moderate-risk genes. And this definitely helps the clinical management of patients. I mean, I'm talking to the choir here at this point. Everybody understands that. Many centers are already testing nearly all their breast cancer patients. But still out in the community, we have NCCN guidelines that restrict to a certain subgroup of patients, okay? And there was a lot of discussion here about population screening. We do have great population-based risk estimates. So that's the average everyday breast cancer, as well as the high-risk family-based ones. So we have two sets of numbers that we can give to the patient, depending on what they look like. And then we're trying to do that for other races and ethnicities. There's a lot more work to be done in that area. What about Hispanics, Asians, Pacific Islanders, et cetera, right? Can we get risk estimates for those people as well? We don't have them today. And then finally, we have VUS at the bottom. I think I've shown you that we have technology now, we have methods now where we're actually going to classify many thousands of these variants in the near term and probably actually solve the VUS problem in the next 3 to 5 years. And I'm going to stop there, but I do want to recognize a number of people. Those in red are the people currently in my lab. These are past members. I really want to recognize Marcy Richardson from Ambry Genetics, who has facilitated all these collaborations that I talked about with their enormous data sets. Megan Holdren helps me to run that HBOP VSEP, so she does great work coordinating that. In the Enigma group, which is the group trying to solve BRCA1 and BRCA2 VUS, we have a number of key investigators here. Alvaro Montero is in the audience here. He's been a leader in this area for many years. The BCAC and SIMBA consortium, Doug and Antonis have done tremendous work, and as I said, that's been going on for 10 years now and continuing. In carriers, that big population-based study, I'm choosing to put Kate and Susan and Jeffrey Weitzel into that category, but we've collaborated on so many studies over the last 25 years, just innumerable studies working together very closely. And then some key collaborators down here, some of whom are in the audience. I want to thank them for their many years of collaboration. I will stop there. Thank you.
All right, Roger gets the first question.
Very, very interesting talk. For the BRCA2 hypomorphs, have you found a different spectrum of phenotypes? And for example, they may be good candidates for things like Fanconi anemia because the patient may survive that kind of hypomorphic biallelic mutation.
Yeah, we really don't have enough clinical data to look at Fanconi, but we have looked at things like age of onset, family history. And as you might expect, their age of onset is later. So they're not your classical BRCA2s at 48 years of age on average. They're actually at 55. So they do come in a little bit later. We're trying to do some studies on tumors now to try and understand the tumors, to see if they've got loss of heterozygosity on the second allele and various other markers like that. Exactly, all of this kind of stuff. We're just trying to do that now. Yeah.
Kate?
Yeah. Thanks, Fergus. I've had the preview of some of that exciting data. I had two questions. I noticed that you didn't see any functional differences in mutations in the tower domain.
Right.
And so I was wondering, that's actually helpful information. And so I want to confirm that. And the second question I had is, you found all these hypomorphs and you found all these missenses, but what proportion of those are actually in ClinVar as being reported in patients?
Okay. Which one do you want to start with?
Do you remember them?
What proportion in ClinVar? I got that much. So what proportion in ClinVar? We actually haven't looked really that carefully because it's changing all the time, right? But I would say maybe 30% are in ClinVar. There's a lot of totally novel that haven't ever been seen in a human yet. But we will over time. As we continue to screen, we're probably going to find a mutation in every nucleotide in these genes. You know, that's fairly certain. The other one was the tower domain. So the tower domain is, if you think about that DNA binding domain, you've got helical, OB1, OB2, OB3. So it's broken into 4 categories, and the tower is in that OB2, the third one down. Oh, it's right here. Good point.
Yeah, look.
We can
right
point
there.
to this. So it's it's this vertical
right
thing
there,
that's
yeah.
sticking up,
Yeah.
right? And so it's interesting. We don't find any pathogenic variants in that region, but we do find about 3 or 4 hypomorphs in that. But they tend to be at the base of it, which probably destabilizes the whole thing. You know, it gets wobbly. We don't really even know the function of that tower. There's none directly in that structure. It's more at the bottom where it can influence how it's bending or folding or something like that.
We'll get Greg Bowman on that. Is he here today? We have a structural biologist who's interested in this. There was a question online, which was actually for our last speaker, but I'm taking the prerogative to address it to you, which is mutation-specific risks. So location of the mutation in terms of risks.
Well,
Can
our
you
friend
address
Dr.
that?
Nathanson is leading a study on exactly that,
Sure,
and
but
has
yours-
done in the past.
But you're standing at the podium, so
I
you
am.
get to answer.
We have found so many years ago, maybe 20 years ago, people observed what's called the OCCR, the ovarian cancer cluster region, suggesting that mutations in the middle of both BRCA1 and BRCA2 had higher ovarian cancer risk and lower breast cancer risk compared to the edges of the genes. That's held up. And we've done probably 5, 6 huge studies with those massive numbers of patients that I just talked about. It's for sure there. And it's about a 1.8 to 2-fold difference. And that's not taken into account when we do risk assessment on patients. We're sitting there with a 2-fold risk. We're trying to use polygenic risk scores that have a 2-fold risk, but we're not incorporating the fact that it's in the wrong part of the gene and there's a 2-fold difference. So I think we have to start building in some of these observations, these biological observations, even if we don't understand what's driving them. Because we don't understand the biology just yet.
Hi there.
Hi, how are you?
Wonderful talk. Congratulations. My question is regarding the phenotypes that you're seeing, if you have any clinical data on the hypomorphs. I know that many of them have not been seen in humans yet, but are we kind of, are the phenotypes, the attenuated cancer risks being seen across the board, or is this some area where there's some mutation or location within gene specificity? For example, might you have a hypomorph where the breast cancer risk is attenuated, but the ovarian cancer or pancreas cancer risk is not? And if we don't have those clinical data, I'm wondering, do you have any guesses as to whether or not there might be that kind of variation, or do we expect it to be an across-the-board change in risk?
I think it's going to be across the board change. They're all in that DNA binding domain, right? So they're not influenced by this OCCR thing that I just talked about. They're outside of that. So they have what you might call the regular risk, except they're only partial effects. We really don't have enough clinical data just yet. A lot of our work has been with testing companies, but they don't have a lot of clinical data, as you know. They just have the bare minimum. So I think once this paper gets out the door and we get more established and we understand the tumors a little bit better, we're going to have to go out and do a sort of a worldwide collaborative collection on these clinical data to try and better understand what's going on there. You know, is it disease specific? You know, is it the same in prostate, pancreas, ovarian, and breast? We've no idea at all. We just barely touching the surface on this stuff.
But it was interesting, right? David Goldgar and your paper on BRCA1 R1699Q, right? Did I get it right?
So that's BRCA1, yes.
Yeah,
right.
but for BRCA1, that hypomorph seemed to attenuate breast cancer risk, but not ovarian cancer risk.
That's true.
So, you know, it will be interesting to see, right?
There's possibilities, absolutely.
Yeah. So getting at the hypomorphs again, sorry, we're just going to pound on that a little bit. You know, it would seem like there's two ways to be hypomorphic, right? One is it decreases lifetime risk, but this interest about PARP inhibitor sensitivity, like I'm actually interested that they were sensitive because there's actually speculation that could be a mechanism of PARP inhibitor resistance, is that the hypomorphs might not respond as well. So were you surprised by that?
I was,
Okay.
because the neutrals didn't shift and the deleterious didn't shift, only the hypomorph shifts.
Yeah.
But it's, I mean,
you
we've
think
done the
that's-
experiment many times and it happens again.
Do you think
That
some,
experimental setting, it's real, but what does that mean in terms of biology? There's a lot more studies to be done to figure this out.
but do you think that some hypomorphs will be they'll be different? That some will
Yeah,
have
because, you know, we say hypomorphs, which means they're all exactly the same. They're obviously not. There's a spectrum, right? So they're probably going from 20% lifetime up to 40% lifetime. And it depends on which hypomorph you're looking at. So there's a lot more details to be figured out.
I was going to just ask about when you think about now this definition of hypermorph, defining which phenotype we're referring to. So there's hypermorphic in terms of the degree to which a mutation drives a disease process, but also that may, as you kind of suggesting, those same mutations might not be hypermorphic when it's related to the PARP inhibitor sensitivity phenotype.
Right.
So how do we start to kind of understand those differences?
A lot more biological studies, right? So we have to look at all the different signaling pathways that BRCA2 is involved by one. We haven't done that yet. This is just, you know, the first observational study. So we know that there's a defect in homologous recombination. That's how we pick them up. We know there's a strong effect of the drug, which is a little bit different. But we do know in terms of risk, where we pull them together in the case control, that they're certainly intermediate and hypomorphic there as well. But there's a lot more to understand. You know, what about replication forks versus double-strand breaks? You know, all these types of questions. Absolutely.
Let's talk about the VUS problem you're solving. So what's the impact of this new knowledge if you know how many actually turn out to be pathogenic and the frequency of these in the general population? You know, honestly, how many individuals are out there walking around who are actually at super high risk? They might have even been told they're at no risk. Like, are we talking about a few dozen people or tens of thousands of people?
So Ambry Genetics went back and looked at that set that we did with them, where we classified from 154, we were only left with 22, so we did the majority. They estimated that 9,000 patients, not probands, but family members included in that, right? But 9,000 people at risk with those variants in them. So that's just that set, you know, so that was 140 or so VUS. We're talking about 439 observed from our CRISPR studies. So, you know, multiply by 3, 20, 30,000 individuals at risk. It's big numbers.
Those are just those tested, right?
Yep.
So,
Yep.
Fergus, just because this was seen in carriers and because it's such an important issue for this group, you mentioned NBN, but I think there's this ongoing thing about the Slavic founder. And so can you comment on both the Slavic founder and CHEK2 I157T and S428F and what you found in carriers?
So these are lots of different definitions. They're pathogenic but lower penetrance. So the Slavic founder is 457del5, I think it was, or del4. We looked at that specifically in carriers. We did have the power. We did have enough numbers to look at it. It's completely flat, 1.0. It just doesn't do anything. So there was a couple of studies out of Poland that published that. Stephen, you might have been involved in some of those. I don't know why Poland was different, but certainly in our large studies, it's completely flat, and I think Bridges have shown the same thing.
We're really getting to the minutia now, but we did find and we did publish that they had to have a second allele on the opposite chromosome in trans.
Okay.
So there was a variant of the NBN gene, which occurred high frequency in Poland, about 45%, low frequency in North America. If you had the mutation and the variant, your risk of breast cancer is increased 3 times.
So there's a subtlety there that we perhaps didn't look at. Yeah. Okay.
There's someone else reported at Will's meeting, a couple more similar situations where there were polymorphic variants which modified.
Right. So
Who,
two
that was
variants
you
together.
or who?
Right. That wasn't us. No.
But it's been showing it up.
Okay.
And sorry,
And then the
CHEK2.
CHEK2 I157T. So this is the classical one. It's got an odds ratio of 1.3. So it's really not clinically significant. You know, it's really low risk. It's like a SNP from the GWAS that I was talking about. But unfortunately, it got labeled in ClinVar and ClinGen in the public databases as pathogenic and confused a lot of people. We don't think of it as clinically relevant. We leave it out of all of our studies. We immediately toss that and a couple of others out the door and try to focus on the higher risk CHEK2 variants.
But I think it's just to hit this point home, they're really common, both of those, and the risk is not high, but they're incredibly common. So we
Right.
see them all
Judy
the time.
Garber had a really nice paper on that last year.
Yep.
Where they tackled the 3 common variants in CHEK2 and showed that they really weren't doing anything important. Yeah.
All right, Bob has another question.
There is a potential of a BRCA1 or 2 mutation that's hypomorphic. And we might go to those individuals and say, wait on your prophylactic surgery, don't. And so suddenly that is a completely different message of urgency that we're bringing opposite, that we're bringing individuals who are found to have a pathogenic mutation. So how on earth are we going to deal with what will appear to be mixed messages on this? In one case, have your surgery. In another case, oh no, don't. So like, where's this headed?
If one thinks about like thyroid cancer, medullary thyroid cancer and RET mutations, there are high penetrance and moderate penetrance RET mutations. And so at least in other diseases, other cancer susceptibility diseases, we counsel that way frequently.
Yeah. And just one point of care there is that this is a functional study. We still need to combine it with all the other clinical data to formally classify these variants with ACMG models, right? That work still has to happen. Jada.
I was just going to build off that and say, I mean, I think to the messaging point, it will probably be received with some dissatisfaction from the community. Some people, however, will probably feel somewhat reassured by the fact that now maybe prophylactic surgery was something they didn't want to pursue or didn't feel comfortable pursuing, and now screening could be a more appropriate option. But I think that with anything, it's so important that we think about this early on and be careful with the way we craft these messages, and probably reassuring people as well that we're not saying don't do anything. Presumably the message will be screening is the thing that could be the right option on the table for you. And so I mean, to me, that's part of the way we deal with the reactants and the fear that people will inevitably
And we
respond
know
to
that
a
some of
message
these
like
women,
this with.
just like with Check 2 and ATM, some of these women will choose to reduce their risk with prophylactic mastectomies. You know, it's an individual choice, after all. We're not prescribing what they have to do. Go ahead, Mark.
I mean, I think we're all sort of riffing on the same thing, but how confident are you that the assay, that something is hypomorphic in the assay is actually hypomorphic in terms of cancer susceptibility? I mean, I was thinking back to the haploinsufficiency work from yesterday and whether if you rolled some of those into that model, would they still have that haploinsufficiency
Yeah,
when
that
they came
work
to,
needs to be done.
fork reversal, for instance?
Certainly for the ones done in the homologous recombination assay, the 1-by-1 assay,
Right.
we're very confident in those, which is the 70 that you're looking at there.
Right.
But then in the bigger MAVE assays, those huge, thousands of variants at the same time, that really still needs to get calibrated.
Yeah.
There has to be a degree of caution. And that's why I was talking about using 2 cut points and only going with the extremes. We really need to be quite careful there.
Also, just to make one point, right now, we're talking about VUSs that are going to be reclassified. We're not talking about things that are currently called pathogenic that we're
Right.
going to be
Yeah.
reclassifying downward. So it just,
Yeah,
it's
so
not, I
everything
think that
we've-
needs to be clear.
All our data here agrees with everything that's in ClinVar already. There's no, so that's an important statement.
All right, this has been great. Any last questions for Fergus? Fergus, thank you. That was terrific. Thank you so much.